Immunology Insights, Basics and Beyond. Chapter 10, T cells. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we delved into the world of B cells and antibody production, exploring how these remarkable cells form the foundation of the adaptive immune response. Today, we are focusing on another essential player in the adaptive immune system, T cells. T cells are critical for orchestrating and executing immune responses, and they come in several types, each with its own specialized function, like helper, cytotoxic, and regulatory T cells. Understanding T cells is crucial for grasping how the immune system not only fights infections, but also maintains balance to prevent overreaction and autoimmune diseases. So, let's talk science. Let's start out with what are T cells? Well, T cells or T lymphocytes are a type of white blood cell that develops in the thymus, hence the T in their name. Like B cells, T cells are part of the adaptive immune system and are essential for identifying and responding to specific antigens. However, unlike B cells, which recognize antigens directly through their B cell receptor, T cells recognize antigens that are presented to them by other cells. Each T cell has a unique T cell receptor or TCR on its surface that recognizes antigens presented by major histocompatibility complex or MHC molecules on the surface of antigen-presenting cells or APCs, such as dendritic cells, macrophages, and also B cells. There are several types of T cells. T cells can be broadly categorized into three main types: helper T cells, cytotoxic T cells, and regulatory T cells. Each of these plays a distinct and crucial role in the immune response. First, we have the helper T cells or Th cells. Helper T cells are the conductors of the immune system's orchestra. They do not directly kill infected cells or pathogens, but instead coordinate the immune response by secreting cytokines, which are signaling molecules that influence the behavior of other immune cells. There are several subsets of helper T cells. Th1 cells promote the response against intracellular pathogens such as viruses and certain bacteria by activating macrophages and cytotoxic T cells. Th2 cells drive the response against extracellular parasites like helminths and are involved in allergic reactions by promoting B cell class switching to produce IgE antibodies. Th17 cells are important for the defense against also extracellular bacteria and fungi, particularly at mucosal surfaces, and are involved in inflammatory and autoimmune diseases. Th17 cells are named after their signature cytokine IL-17, which drives the recruitment and activation of neutrophils and also induces the production of antimicrobial peptides, as well as enhances the barrier functions of epithelial cells. T follicular helper cells provide help to B cells in germinal centers, facilitating their activation, class switching, and affinity maturation. Helper T cells are activated when they recognize an antigen presented by an APC, in conjunction with MHC class II molecules. Once activated, they proliferate and differentiate into various subsets, secreting cytokines that shape the immune response. The second main T cell subtype are cytotoxic T cells or CTLs. Cytotoxic T cells are the immune system's assassins. Their primary function is to kill cells that are infected with viruses or have become cancerous. Cytotoxic T cells recognize antigens presented by MHC class I molecules, which are found on almost all nucleated cells in the body. When a cytotoxic T cell recognizes its specific antigen on an infected or abnormal cell, it releases cytotoxic granules containing perforin and granzymes. Perforin forms pores in the target cell's membrane, allowing granzymes to enter the cell and trigger apoptosis or cell death, effectively killing the cell and stopping the spread of the infection. Cytotoxic T cells are crucial for controlling viral infections and eliminating cancer cells, making them a key component of the immune system's defense against these threats. The third main type are regulatory T cells or Tregs. Regulatory T cells are the immune system's peacekeepers. Their primary role is to maintain immune tolerance and prevent autoimmune diseases by suppressing the activity of other immune cells that might otherwise attack the body's own tissues. Tregs are essential for maintaining the balance of the immune system, ensuring that the response to pathogen is strong enough to clear the infection but not so strong that it causes excessive damage to the body's own tissues. They achieve this by secreting anti-inflammatory cytokines such as IL-10 and TGF-beta, and by directly interacting with the other immune cells to suppress their activity. Regulatory T cells are crucial in preventing autoimmune diseases where the immune system mistakenly attacks the body's own cells, as well as in controlling chronic inflammation and maintaining tolerance to harmless antigens. Now, let's look at T cell activation and differentiation. The activation of T cells is a highly regulated process that ensures the immune response is both specific and appropriate to the threat. For a T cell to become activated, it must receive signals from its TCR binding to an antigen MHC complex as well as co-stimulatory signals from the APC. Once activated, T cells undergo clonal expansion, where they rapidly divide to produce a large population of effector T cells that can fight the infection. These effector T cells then differentiate into their respective types, helper, cytotoxic, or regulatory, based on the signals they receive from their environment. The differentiation of T helper cells into their specific subsets, so Th1, Th2, Th17, and TFH, is influenced by the cytokines present during their activation. For example, the presence of IL-12 drives the differentiation into Th1 cells, while IL-4 promotes the differentiation into Th2 cells. Like B cells, T cells can also form memory cells after an infection has been cleared. Memory T cells persist in the body for years, ready to respond quickly if the same antigen is encountered again. There are two main types of memory T cells: central memory T cells, which reside in lymphoid tissues and can rapidly expand upon re-exposure to an antigen, and effector memory T cells, which circulate in the blood and tissues, ready to mount an immediate response. Memory T cells are crucial for a long-term immunity, ensuring that the body can respond more effectively to pathogens it has encountered before. This is also the basis for the effectiveness of many vaccines, which aim to create a pool of memory T cells that can respond quickly to future infections. T cells, however, play also a dual role in health and disease, and their dysfunction can have profound consequences. HIV, for example, specifically targets CD4 positive helper T cells, weakening the immune system and leaving the body vulnerable to infections and cancers. Overactive T cells, on the other hand, are implicated in autoimmune diseases, such as multiple sclerosis and type 1 diabetes, where the immune system mistakenly attacks the body's own tissues. T cell-related cancers, such as T cell lymphomas, arise from abnormal proliferation of T cells, reflecting their importance in immune regulation. Additionally, T cells are central to graft versus host disease, or GVHD, a serious complication of bone marrow transplants where the donor's T cells attack the recipient's tissues. Conversely, in host versus graft reactions, the recipient's T cells reject transplanted organs. These diverse conditions highlight the critical balance T cells maintain in immunity and tolerance. Given their central role in the immune system, T cells have become a major focus in the development of immunotherapies, particularly cancer. One of the most promising approaches is CAR T cell therapy, where the patient's T cells are genetically engineered to express a chimeric antigen receptor, or CAR, that targets specific proteins on a cancer cell. These modified T cells are then infused back into the patient, where they can seek out and destroy cancer cells. CAR T cell therapy has shown remarkable success in treating certain types of leukemia and lymphoma, and research is ongoing to expand its application in other cancers. In addition to cancer, T cell-based therapies are being explored for the treatment of autoimmune diseases, chronic infections, and even in the context of organ transplantation, where regulatory T cells may be used to promote tolerance to transplanted tissues. To conclude, T cells are the orchestrators, executors, and regulators of the immune system, playing essential roles in both fighting infections and maintaining immune balance. Their ability to adapt and respond to a wide range of threats makes them indispensable in the body's defense mechanisms. As we continue to unravel the complexities of T cells, their therapeutic potential becomes increasingly evident, offering new avenues for treating diseases that were once thought to be incurable. In our next chapter, we will cover one of immunology's founding figures, Eli Mechnikov, whose discovery of phagocytosis revolutionized our understanding of innate immunity. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.